I'm Helen O'Neill, and I am speaking at my local time here, which is 8 p.m. in London. It is a hot, hot evening in London. I am not built for this heat. I am really looking forward to today's topic. I will be moderating the session along with the second best moderator, Barry Behr, who is usually from Stanford, but where are you in the world now? You're on vacation.
I'm traveling across country. I'm outside of Indianapolis, Indiana at the moment.
Wow, that's amazing. Professor Behr is a clinical embryologist who graduated from University of Nevada, Reno with honors and earned his PhD with emphasis in reproductive physiology. I'm not sure what that means, but I'm sure it's important. He has published so much that if I read it all out, you would probably all leave. So we'll mention his multiple awards, zillion presentations, and we'll just end with the fact that he's a major thought leader in clinical embryology. Over to you, Barry.
Well, thanks so much, Helen. It's great to work with you virtually on this fantastic platform for learning and sharing information. I hope I stay connected with you here with my mobile bandwidth, but I'm happy to be working with you and being the second best moderator. But let me tell you a little bit about the world, a little bit about you, Helen, who I just recently met as well. Dr. O'Neill is a lecturer in reproductive and molecular genetics and director for the MSc in reproductive science and women's health at the Institute for Women's Health at the University College of London. Her research focuses on pre-implantation embryo development and the use of genome editing to assess understanding and the treatment of disorders of infertility, not surprising, what we're here today to learn about. Dr. O'Neill has an honors degree in genetics, an MSc in prenatal genetics and fetal medicine, and she lectures both to master's and medical students and is active in public engaging involving genome editing. And if that's not enough for you or for anybody, she's also the CEO and founder of fertility health and precision medicine-based approach to reproductive health. She's, most importantly, a mother of a one-year-old, which I'm sure keeps her very, very busy.
She did not want me to give this talk tonight. She was very, very adamant about that.
I'm sure. But I think before we begin, I think we are all here because we realize how important and how dynamic the genomics and genetics field is and how it relates to reproduction and how powerful these tools can be in changing future generations. And I'm very excited and honored to be co-hosting this platform with these outstanding speakers. Helen, would you like to tee it off from here?
Great. We have a very talented team, just so as you know, who are working behind the scenes to post your questions. So we encourage you to all post your questions using the Q&A section of the conference service and not on the chat function of Zoom, please. Okay, I am going to dive straight in and introduce our first speaker, Jamie Metzl, who is galactically known. Jamie is a Renaissance man, and has an amazing career. He is a technology and healthcare futurist, a geopolitical expert, politician, novelist, entrepreneur, and media commentator. He is senior fellow of the Atlantic Council, Singularity University faculty member, and the founder and chair of One Shared World. He serves on the World Health Organization Expert Advisory Committee on Human Genome Editing. Jamie previously served at the US National Security Council, State Department, Senate Foreign Relations Committee, and as a human rights officer for the UN in Cambodia. Jamie appears regularly on national and international media and his syndicated columns, among other writing on science, technology, health, and politics. There's about 4 other lines here on you, and I'm going to skip ahead to say, Jamie, welcome. Fantastic bio. We're looking forward to hearing your talk.
Well, thank you so much, Helen, and thank you, Barry, and thank you to all of you. It's really just a pleasure and an honor to be here with you. But I just have to make one point of order. Helen called me a Renaissance man, and I just want to make clear that about 99.9% of everyone who lived in the Renaissance was shoveling manure. So the question is not Renaissance, but where in the Renaissance? And each person can imagine themselves. So I hope that I will prove a little bit of what we're all here today exploring, which is that to understand the future of reproductive medicine, we need to start with a wider aperture, to think about where is society going, where is technology going, and then the future of reproduction and of reproduction medicine must sit within that framework, because what we're experiencing now is so profound that if we don't think broadly, we're going to miss it. We aren't going to be able to prepare for the world that is coming, and it's coming so rapidly. So when we talk about reproduction, it's pretty ingrained. Sexual reproduction is pretty ingrained in our and in other species. But we need to start from the point of it's kind of a relatively new phenomenon. Life's been around for about 4 billion years, and we're only talking about a 1.2 billion year history of sexual reproduction. So if we're talking about changing it, at least it's relatively new compared to all of life. So the good news is that we humans are a sexually reproducing species, and whatever happens with technology, we're going to remain a sexually reproducing species. The maybe bad news is we may not be sexually reproducing through the act of sex. And I think that's a pretty big and profound transformation that is just beginning to play out. I mean, many of you, like me, have been celebrating these Louise Brown birthdays every year for the past 42 years. And so we know that this idea of sexual reproduction that happens in many ways outside of the human body, it's new and it's still experimental, even if it's becoming more common. But to think about, as I was saying, where this future is going, we just need to take a step back and assess where we are now and where we're headed. As all of you know, when Watson and Crick and Rosalind Franklin and others identified the double helix structure of DNA in the 1950s, what they were doing was identifying, in essence, the book of life, and the form of the book of life in this twisting ladder. And with the advent of genome sequencing and all of the various iterations of genome sequencing, we've shown that the book of life can be read. And now we've had for decades, we've been in the era of human genome editing, but now with the new tools, CRISPR and base editing and many others and many more to come, we are in the era of writing our genetics. And when we think of technologies that are readable, writable, and hackable, our minds go to IT. And we've internalized that IT is always transforming. And if you get a new iPhone and it's the same as your old iPhone, you don't think, oh, that's how technology works. You think, wait a second, we've passed beyond that experience. We need something better. We need upgrades. And that's what's exciting and what's frightening about this moment is that our one little species among these billions that have ever lived are now having the capacity to do upgrades. That's not something new. We've done, anybody who's immunized or has a pacemaker or frankly lives in civilization is a beneficiary of past upgrades, but our capacity to do that is increasing. And one of the reasons that it's increasing is we're seeing a super convergence of all of these technologies. Everything that we're talking about today rests upon multiple revolutions, whether it's the computer science revolution, the computing revolution, AI, machine learning. So many of these technologies they all fold into each other. And that's why our experience of technology for centuries, but certainly now, is accelerating at an exponential rate. And it's also why when we think about where these technologies and where we are all heading, we have to try to overcome the conservative bias of our brains, which has to do with our evolutionary history and the needs of survival in the African savannah. But we really need to, in many ways, think like science fiction writers, because the boundaries between science and science fiction are really blurry. And when we think about how these technologies, certainly in the fields of genetics, biotech, and reproductive medicine, are going to push forward, the application that's nearest to what we're all doing now is what Helen is going to talk about in just a moment. And that is this transition from generalized medicine based on population averages. So traditional cancer treatments, you go and you have a cancer and they say this looks like a X cancer and we'll try this treatment. If it doesn't work, we'll try something else. And if that doesn't work, we'll try something else. To our world of precision medicine based on each person's individual biology. And in order to understand each person's individual biology, we need to know who they are. And that is through all kinds of family histories, biometric information, but perhaps the most important piece of information over time will be everybody's sequence genome. And that's why we're going to relatively quickly move toward a world where everybody is going to have their genome sequenced just as a foundation of your electronic health records and of your basic health care over the course of life. And if it doesn't happen before you're born, it'll happen when you get your heel prick blood test as a newborn, because you're going to need it. And so when that happens, and I think that will happen quickly, we will very quickly get into a world where millions and then billions of us will have our whole genomes sequenced, and the cost of whole genome sequencing is trending towards negligibility. But we'll also need to have the information about people's lives, the phenotypic information that will come through their not just electronic health records, but electronic life records. And when we put together those two things, these massive databases of genetic information with life information, very quickly, we're going to be able to use our tools of machine learning and big data analytics to crack the code of complex genetics and systems biology more broadly. We're going to hear from Nathan in just a little bit about polygenic scoring. And that's polygenic scoring is hugely important because we're moving from this early stage of genetic analysis where we can understand these single gene mutation disorders to understanding the predictive power of complex genetics, which are different for everybody. And so we really need these massive, massive data sets. And there's a theological question. I recently had a paper published by the Vatican talking about the Catholic Church and the future of human genetic engineering. But it's a big question. If you believe, as I do, that human beings are single-cell organisms gone wild over 4 billion years of evolution, ultimately we'll be able to understand humans about as well as we understand single-cell organisms today, which is not perfect, but it's certainly a lot more than we can do now. So when we move from our simple understanding of genetics to our complex understanding of genetics, because of this massive increase driven by precision medicine, that's going to change the way that we do a lot of things. One is that we're going to realize that a lot of what we do and what we experience is somewhat predictable, whether it's the disease states, as Helen will talk about, for genetic disorders, but also things like personality style, certain things that we're good at, maybe things that we're bad at. We're going to have all of that kind of information. And that's going to, one, transform the way we think about genetics, and it's going to move it out of the realm of healthcare. Because right now, some people, you can send in your mouth swab to 23andMe, but you get a little bit of information. When we think about genetics, we think of it in the context of healthcare more than we think about it in the context of parenting and thinking about our future life or career choice or partner choice or all kinds of these very, very tough things. But the second thing that is going to transform is the way that we do baby making, not just the way we make babies, but the nature of the babies that we make. Because human reproduction comes with an error rate. We don't call it an error rate, we call it a random mutation, which is part of our evolution and survival process. But it means that some people have mutations that give them extra powers, and some people have mutations that cause them to die horrible early deaths. And we're going to have a lot of this information. And when we get to the point where science has a lower error rate than nature, most everybody is going to want to reproduce through science rather than take the risk of reproducing in the back of a car or in a hovercraft or in any way that's just relying on the systems and structures that nature gave us. And I know that's scary to people, but nature didn't give us houses or parkas or air conditioning or heating or immunizations or medicine or agriculture or all those things. So it shouldn't be as scary as it is. That doesn't mean it's not complicated. And so what we're going to see is an increased adoption of first IVF and embryo screening, which, because this is a group of experts, IVF and pre-implantation genetic selection, which will start in the early stages as it is now for high-risk pregnancies and for screening out terrible disorders, but relatively quickly, because we're going to have so much more information of the genome and understanding of the story that complex genetics are telling us, more and more people are going to want to use embryo screening as a mechanism for selecting against certain things, going from simple to complex single gene mutation disorders, but then it will be complex things that people are afraid of. But because there's no natural boundary between normal and abnormal or therapeutic applications and enhancement, it's going to be impossible to police a fine line between these different uses. And there's just so many. How bad of eyesight is too bad? How short is too short? How short of a life is too short of a life? We all exist on a spectrum, and we're going to have to live in that spectrum. Also transforming the way we reproduce will be the use of iPS cells, induced pluripotent stem cells, to predominantly increase the number of eggs. As you all know, eggs are far more scarce than sperm, but if we can increase the number of eggs using iPS cells to induce an adult cell into a stem cell, into an egg precursor cell, into an egg, suddenly we can have 10,000, 100,000 early stage embryos, which we can analyze using in an automated way, using the same tools that we're now using for Pcos. And I see that as a primary driver. I think that we're going to talk about the genome editing of pre-implanted embryos in a moment, but embryo selection is going to feel much more comfortable to most people than editing. And then we have this new era of genome editing of pre-implanted embryos. If we had been speaking here a few years ago, we would have said, oh, maybe this is a decade off, but everybody knows what happened with He Jiankui that propelled us into the era of genetically modified embryos. So we are there now. Certainly that will continue. We haven't reached the safety threshold. I'm part of the WHO International Advisory Committee on Human Genome Editing, and we've been very clear, others have been clear, that we're not there yet, but we will be. In spite of the articles that have come out earlier this year about the kind of chromosomal mayhem coming from CRISPR, I think we should just assume that we will get to a level of efficacy where small numbers of genome edits can, and I think almost certainly will be made. And then there's the question is, will we have the capacity, and this is an open question, to go from single gene mutation, from fixing single gene mutations that would otherwise cause terrible deaths where the cost benefit analysis is relatively clear, to doing things that are more complicated, especially because we understand so little of how life functions, that the more changes that we make or seek to make, the greater the probability that we're just going to make mistakes, well-intentioned mistakes, but because we don't fully fathom the full complexity of these systems that we're trying to change. And certainly there are people, George Church's labs, Dieter Egli at Columbia, and others who are asking the question, well, can we do the genome editing at the egg and sperm precursor cell level? And if so, or even at the egg and sperm level, that would open up other possibilities of lower risk interventions. All of this is to say is that we are heading very, very rapidly into this new world. It's cheating to call it a brave new world, because it's scary in some ways and it's exciting in some ways. As I said, our biology is buggy, and we shouldn't be scared about fixing those bugs. It's the history of our species, but we need to recognize that this is really tricky stuff. We're touching on the stuff of life. We all come from different cultures with different norms and expectations. There are monumental ethical issues of equity, of diversity, that we may all end up making choices that seem perfectly logical to us, even in the name of health, even in the name of what we see as survival, but those could end up actually being harmful decisions for us collectively. And how do we navigate the boundaries between individual choice and societal interests? And there aren't easy answers to that, and certainly our WHO committee is struggling with this. But one thing is for sure is that the people in the ART industry, you are going to be the front lines. Whatever, people are going to come to people in your community with these questions. And some of these questions will go way beyond the actual your role or how you see your function, but people don't have another place to go. There are all these, that's certainly why I wrote Hacking Darwin, and there are lots of great resources out there for people if they want to understand this story, but most people aren't going to do that. They're going to come to you and they're going to have a lot of questions, some of which will have something to do with the work that you're doing day to day, and some will be much broader. And even more than that, all of these questions are going to have to exist within the broader context of how we are organized nationally and internationally. I mean, the UK, in my mind, probably has the best regulatory infrastructure in the world, and they've thought a lot about these kinds of questions. In the United States, we're stymied by the abortion debate, so there are a lot of really important conversations that we can't have. And in the world, there are some countries that are very well organized and some countries that have nothing, and we're going to see this kind of reproductive tourism, just like we see tourism for stem cell treatments and other things. And for all of this, what seems like an individual decision for a person, what seems like a conversation between, let's say, prospective parents and their providers, is going to have to plug in to a much bigger story of how we as a species are handling these new, in many ways, godlike technologies that have the capacity to fundamentally transform our species, but maybe for better and maybe for worse. And so that's why I was so appreciative when Jacques invited me to speak to this group, because I know what I'm saying sounds a little bit like sci-fi, and there are some people who are saying, well, wait a second, you know, my job is to look into a microscope and make a best guess about which embryo should be implanted. But all of this is part of this bigger story. And all of this is part of a story that feels like science fiction, but that science fiction is becoming one of the essential realities of our lives and our futures. So let me stop there.
Thank you very much, Jamie, for that outstanding, thought-provoking presentation. While Helen gets teed up for her presentation next, I wanted to ask you just a couple of quick questions. You directly stated, more than alluded to, maybe the discrepancies that may exist in different societies and different cultures. But thinking of Western or capitalistic societies, when do you think healthcare systems will require some kind of sequencing or preconception genomics to ensure that we have less morbidity and less suffering and less expense on the healthcare system?
Yeah, it really depends. So in Northern Europe, it's that everybody has to have this prenatal screening. And because of that, it's paid for by the government within the national health plans. That's why we're seeing almost an elimination of Down syndrome, for example, in Northern Europe. And there's going to be this very, very difficult negotiation because on one hand, in my mind, that's why I said we're going to move toward the end of sex for reproduction, because societies are going to want those choices to be made. but parents will want them to be made even more. And it's also going to be free over time, because if you add up the total expenditure on paying for treatment of people who are born with, in many cases, deadly and genetic disorders, I mean, it's a huge amount of money. I quantify the numbers in my book. So society is going to have an interest in that. But this is really sensitive stuff. What we're talking about, we can't be glib about it. This is the future of life. And people come from different traditions with different philosophies. And I wrote about that a lot in my piece for the Vatican. And so I think it's going to happen in different places at different rates. I would say northern places like Northern Europe, China, Japan, Israel are going to be first movers. But societies change. I mean, there are lots of people who are born with terrible things where we blame the parents for not being careful, whether it's because of fetal alcohol syndrome or other thing. And so I do think that our norm of what is required of a parent to have healthy offspring is going to change, and that's going to push governments.
Perfect. Well, thank you for that opinion, and I think it's very valid, and I'm looking forward to being young enough to see some of this happen, hopefully in my lifetime. So thanks, Jamie. Again, I look forward to you participating in the question answer period where the rest of the attendees can contribute and ask some questions. So Helen, if you're ready to share your screen so we can hear about precision medicine in precision health in reproduction. So Helen, take it away. Thank you very much. You just unmute, Helen.
Thank you. Thank you so much. Thanks for the introduction and for a great talk, Jamie. It's really exciting to see what's coming and to, I guess, field our way between what's real and what's not real. I am talking about precision medicine in reproduction, and I firmly believe it is time for prediction and prevention. So here are my disclosures. I am CEO and co-founder of Fertility Health, which is a proactive reproductive health test for women. I want to make it very clear at this point that my own academic research involving genome editing and my company are very separate entities and do not overlap in any way. They both fall under my passion, which is precision medicine. So just to give a brief overview of my talk, I'm going to talk about what precision medicine is, how precision medicine can be applied to reproductive medicine, how it can be applied now, and what it might look like in the not so distant future. I'm going to call on the big need for big data and just have a brief overview of what the future might look like that's maybe set in reality and not dystopia. So we are all very familiar with prevention is better than cure. And in an ideal world, we would prevent illness, but in the real world, we cannot always prevent. But it doesn't always have to be an either or situation. In order to prevent, first, we must predict, and where we cannot or have not predicted, then we must find ways to cure. But our ability to do the former is advancing rapidly through precision medicine. And I'm going to be talking about the former, not the latter. Until next time where I talk about CRISPR. So precision medicine, or P4, is also known as personalized medicine. Now the term P4 or P4 medicine was introduced 12 years ago by a molecular biologist actually and an oncologist, Leroy Hood. The 4 Ps, predictive, preventative, personalized, and participative, represent the cornerstones of a model of clinical medicine. If we can predict, then we can prevent. However, in order to do so, we need participation from the patient or person if we can prevent them from becoming a patient at all. Now, these 4 paradigms offer concrete opportunities to modify the current healthcare paradigm. Here we see the past, which is based on intuition medicine, signs and symptoms, how does something look? The present, which is set in evidence-based medicine and clinical trials, and the future, which sees precision medicine and algorithms taking a hold of what we are currently using for our practice. But what about precision medicine for reproduction, or precision reproduction medicine? reproductive medicine. There are another 4 Ps to address here, not forgetting prediction, prevention, personalization, and participation. And these are period health, pre-contraception, prenatal, and postnatal, each of which requires prediction, prevention, personalization, and participation. Here's an unpopular notion. It may not sit well with the majority of you. The field of assisted reproduction actually thrives on lack of participation or prevention. It's arguable that it thrives on the other 2 as well. So I want to talk about what we can be doing to prevent the infertility that can be that is caused due to avoidable pathologies. And I include age-related fertility decline as the most predictable pathology. So in order to measure our ability to predict, we can quite easily look at the incidence of certain pathologies, common pathologies, and we'll work backwards from there. But before I do that, let's look at just some very simple facts that are well known and familiar. These are common statistics in reproductive health, and these contribute to infertility. PCOS. 1 in 3 to 4 women have PCOS, which is a known effect, has a known effect on your fertility. It takes on average 5 years to be diagnosed in certain places. 1 in 10 women suffer from endometriosis, which has, again, a significant effect on your fertility. The average time for diagnosis with endometriosis is 7.5 years. 1 in 3 women, we are lucky, will suffer at some point in their lives due to a gynecological pathology. Other known causes of female infertility include ovulatory disorders, chromosomal abnormalities, tubal blockage, and the biggest contributor to this is age. As we know, the mean age of a first mother has gone from being 22 years old to 29 in some countries, and it is increasing. But what can we do to improve the odds of natural reproduction? It doesn't really take much to realize there's a problem when you try add these statistics up in a general population and recombine them in any way. It's really not surprising that we are seeing fertility rates of 1 in 6. After all, we don't need algorithms to predict age-related fertility decline. This is shown to be devastatingly plummeting after the age of 30 on this graph. Nor do we need algorithms to predict the incidence of age-related rise in aneuploidy, which is tragically growing exponentially on this graph in orange. What we need is robust data that speaks to the outliers, the 24-year-olds with premature ovarian insufficiency and the 28-year-olds who have recurrent pregnancy loss due to aneuploidy. We need an accessible triage system which red flags these women before it's too late. What we need is robust stratified data. Another name for precision medicine is actually stratified medicine. Stratified medicine is essentially an approach which stratifies patients based on their own disease subtype. It stratifies them based on their risk, their prognosis, their treatment response, and it does so using specialized tests or diagnostic tests. The key is to base all medical decisions on individual patient characteristics, including molecular, behavioral biomarkers, rather than these population averages, so that we can start to speak to these outliers. The current data sets in assisted reproduction rely heavily on women presenting from infertility clinics or gynae clinics. We need more data from healthy females. We need more data from seemingly healthy females. Personalized medicine is deeply connected to and dependent on data science, specifically machine learning, also known as artificial intelligence more popularly. And while in recent years there's been a lot of enthusiasm around machine learning and the potential of big data, actually there are only very few examples which exist in the real world that actually impact current clinical practice. Now, it is this lack of impact on clinical practice that can largely be attributed to insufficient performance of algorithms, the difficulty in interpreting data, complex model predictions, and lack of validation across prospective clinical trials in order to demonstrate a clear benefit compared to the standard of care. We are conducting a clinical trial at Fertility Health, which is collecting the largest data set of female gynecological pathologies. We are doing this to assess the potential of state-of-the-art data science approaches for personalized reproductive medicine. Our clinical trial is running out of 4 different gynae clinics: PCOS, endometriosis, pelvic inflammatory disease, and recurrent pregnancy loss. And we are aiming to train our algorithms to predict. And we want to do this so that we can better prevent. In order to do this, we clearly need participation. I've mentioned the 3 Ps. Hence why we've made this commercially available. In order for this to be widely available, widely adopted, and for us to get significant numbers for population-based stratification, we have essentially made this an at-home test, which is commercially available, which uses a clinically curated algorithm, which is a questionnaire which has an internal symptom weighting. And embedded within this, we have embedded an algorithm which will enable us to tailor or personalize, I've mentioned the 4th P, each hormone test to every woman. What we're hoping to do is attack the 4 Ps from precision medicine for the 4 reproductive Ps. To galvanize our findings, we aim to combine biometric data, hormone data, genetic data, and let's not neglect reproductive history in order to enable accurate prediction. Now, these are the 4 Ps that I mentioned in reproductive medicine, and I'm going to quickly touch base on all of these just so we can get a better idea of how we're using personalized medicine in reproduction. Starting with period, we've actually seen that widely available period tracking tools or apps have been challenging the dogmas of reproductive science, but they're challenging the average cycle length by showing that actually using real-world data, the mean variation of cycle length is different in a woman who has a higher BMI than that of a woman with a lower BMI. As you can see from big data, stratification is key. Moving on to preconception, what I want to talk about is the use of genomics and how we can embrace genomics in all of these areas to better understand how we can move forward for prediction. And really what I want to do is some last minute thought provoking. It's difficult to cover all of these topics and what is happening in the genomics world across all of these, the life cycle of reproduction, and perhaps throw in some last minute discussion points about what we are doing and what we can do, and maybe put a little bit of context about what we're already doing. Far too often we dwell on technology or new technology and assume that that we are going to be using it for different purposes, when in reality, we're using it for the same purpose. In reality, we have an improved ability to do what we're already doing, but having an easier way of doing this often incites fear in people. It always invites the bioethicists to the room, and definitely gives a shout out to the dystopia dwellers. So I'm going to start by touching on preconception care and preconception screening. And what I really want to talk about with preconception is why it is that we aren't using genomics for finding the other folic acids. If we know that folic acid can reduce the cause of neural tube defects by 70%, why are we not investing more in discovering other folate type substances which could be reducing the genetic burden through simple dietary means? Yes, I'm used to talking about genome editing, but there are far more simple means out there that we can be used in the preconception and prenatal stages. As for prenatal, or as for preconception carrier screening, if 1 in 3 people are carriers of a recessive condition and 8 million babies are born with a congenital disorder, why is routine carrier screening not so routine? Why is it frankly not more routine? After all, both mandatory and optional screening programs have seen major successes for previous, in other countries, for cystic fibrosis, for the hemoglobinopathies. The reality is we just need to make it more equitable. We need to make it people more educated. We need to make it more accessible. As Jamie mentioned, with the fall in whole genome sequencing, this is entirely possible. Prenatal screening has benefited hugely from the use of non-invasive prenatal testing and diagnosis. Perhaps it could do with being more refined, but our means of using cell-free DNA are amplifying our methods of detection and prediction across the board. In the postnatal stage, I'm going to end with a more controversial aspect, which I do not find controversial myself, and that is newborn screening, and the idea that we should screen every newborn and have the whole sequence of their genome. To continue with the folate analogy, if simply avoiding phenylalanine in a newborn can prevent mental retardation caused by phenylketonuria, what are the other phenylalanines that are causing all of these other metabolic disorders? At the moment, certainly in the UK, we only screen for 7 to 9 metabolic disorders using the Heel-Prick test, when there are 700 inborn errors of metabolism, which could easily be prevented if we invested more in genomics, genomic prediction, not the company, and our ability to decipher natural compounds that could have a preventative effect on these underlying and devastating pathologies. I will end by saying that genomics is transforming reproductive health. I look forward to presenting our findings from the big data in future meetings, and I will end by taking any questions at all. Thank you for your time.
Thanks so much, Helen. We're going to save more questions to all the panelists in a few minutes, or a little bit in about 20 minutes or so after Nathan's presentation. So while his presentation is being teed up, I thought I'd, well, first of all, compliment you on an excellent presentation, and then ask you how you think ethnicities play into some of these precision health questions. I mean, we always wonder, how does this, condition apply to me? Or I smoke, I'm not going to get lung cancer. It doesn't happen to me, it happens to somebody else. And then there's also a lot of these algorithms and bioinformatics things are based on certain populations. So I'm just wonder how you address different ethnicities in some of these precision health or personalized medicine approaches.
Essentially what we need is more genomic data. We need sequencing across every ethnicity. We know certainly in reproductive health that Black women are more prone to fibroids. We need to build this into our factoring for prediction of their infertility and how they're going to be affected by this. When it comes to whole genome sequencing, our reference genomes, we need to have reliable reference genomes. We cannot rely on a reference genome which was sequenced back in 2000. We need to have new reference genomes, relevant reference genomes, not those based on the Ashkenazi Jewish population, because the chances are we'll get more hits on the mutation load, but on every individual. And as I said, the cost of sequencing is dropping so significantly that there's no excuse that we don't have a decent control.
Yeah, well, that's spot on. Thank you for recognizing that. I think it's really important, and hopefully we'll get more data soon.
Our analysis is only as good as the reference.
Okay, so I think it's you have the mic to introduce Nathan next, I believe.
Fantastic. Nathan, are you there?
Yes, I am here. Thank you.
Great, great. Nathan, I'm going to go straight in and give you a background about Nathan Treff, who received his PhD in biochemistry in 2003. He performed postdoctoral fellowships in embryonic stem cell biology and reproductive genetics at the University of Wisconsin. He is currently Chief Scientific Officer of Genomic Prediction and ReproCare Genetics Scandinavia. He's board certified in molecular diagnostics as a high complexity laboratory director, an associate professor at Rutgers University, and a senior editor of fertility and sterility science and JARG. Dr. Treff has published over 100 peer-reviewed papers in reproductive genetics and has served as a member of the ASRM Research Committee for 5 years. His work is focused on development and validation of comprehensive genome analyses in the pre-implantation embryo. For his research, he was awarded 6 prize paper awards from the ASRM in 2013 and Esther Rosenwach's Young Investigator Award and the 2013 Bonai Olam Scientific Achievement Award. He was also awarded with a 2014 EMD Sorono Grant for Fertility and Innovation. He has recently published work on PGT for polygenic risk scores and has been reported on the Wall Street Journal, Netflix, Forbes, The Economist, and Here Tonight. Most importantly, Nathan, I'm really looking forward to your talk.
I'm excited to talk about the testing that we've developed, starting to look at polygenic risk scoring in the human embryo. I'm currently a shareholder and co-founder of Genomic Prediction, and I will be talking about products that we've developed today. I'll start by giving an introduction into Genomic Prediction. I'll discuss our new platform called LifeView for PGT, how we've expanded PGT into polygenic risk scoring, and practical applications of this technology. Our mission is really to help would-be parents and give them more information so that they can give their children the best possible start, providing them with the power of choice over chance. Our company is made up of several important individuals. My co-founders, Laurent and Steve, really pioneered the development of polygenic risk scoring to look at genetic disease using the genome and machine learning algorithms. We also have Jia Xu, who's leading our technology development. She's originally from IBM Watson Genomics, as well as Jennifer Eccles, who has 20 years of experience in genetic counseling, and our head of operations, Leslie Duffy, who's also been involved in the industry for over 13 years. We are located at the New Jersey Bioscience Center, and we have accreditation for the testing I'm going to be talking about, both from CLIA and CAP. The LifeView PGT platform is one of the highest throughput methodologies in the field. We look at over 800,000 SNPs, and this really gives us a very high resolution of looking at the genome, kind of like going from a standard definition television to a 4K TV. And one of the advantages I'll talk a little bit about is that we can directly test mutations as opposed to just using linkage-based approaches. And then one of the reasons we use this is because it's commonly used in biobanks, which is important for being able to use these machine learning algorithms to predict polygenic disease risk. And this is just, it's basically just one platform. And so from one biopsy, we can obtain results for all 4 of the major tests. And I think one of the main advances we've made is to be able to combine both copy number and genotyping data. What's shown here is concordance for the 4 major areas of testing. And normally these involve independent analysis of copy number and genotyping, but we've actually integrated the 2 into the algorithms used for this platform. Many of us have seen the typical copy number plots that we might get from commercially available next generation sequencing platforms. And what I'm going to show you is what we can actually do beyond copy number by having genotyping information. This is what we call a Violin Plot, and what it shows is an allele ratio. Normal chromosomes have a 50-50 ratio of heterozygous SNPs. But you can see for chromosome 16, this shifts to a 2-to-1 ratio, and this is indicative of trisomy. And you can actually see a pretty clear difference even without the use of copy number information. This is genotyping data only. Here's an example of monosomy of chromosome 4. And when we look at the entire genome, we can actually detect things like triploidy, where there are 3 copies of each chromosome, as well as haploidy. And while this might not seem to be important in terms of the frequency, we've really only started to look at it in more detail. And so far in our patient population, about 1 in 150 blastocysts has either triploidy or haploidy. And again, this kind of diagnosis would be missed by copy number methods. We also have detailed analysis at the chromosome level. So what I've shown here is just a genome-wide plot. If we actually take this individual chromosome and look across the entire length of it, this is an example of data on chromosome 16. Again, you can see the 2-to-1 ratio across many positions on the chromosome. And we can also determine the parental origin of aneuploidy. In this case, you can see that the extra allele comes from the maternal genome. And we've also introduced the ability to predict the cell division origin of aneuploidy, where for example, the maternal DNA, you can see, contributes 2 different alleles, meaning that this is meiotic in origin, and this may help to improve the specificity of diagnosing mosaicism. So for example, if you see an intermediate copy number, but it's meiotic, it's probably not mosaic. And again, this genotyping data is missed by copy number. With PGTSR cases, normally laboratories can distinguish between embryos that are either normal or carrier or unbalanced. With the genotyping, we can distinguish whether or not they're normal or carriers of the balanced translocation. And again, this is information missed by copy number. So that's one example of something that we can actually do that many can't, and that actually leads to many laboratories sending us these cases. And so we have an unusual population of patients where these difficult cases get processed. I mentioned the resolution of testing. This is an example of an embryo, and it's chromosome 15 derived from a couple where the father was a carrier of a translocation. And you can see within this segment the 2-to-1 ratio indicating a gain of material, and that the material was gained from the paternal genome, which is expected given that the translocation comes from the father. And we know that it's meiotic in origin, and you can see both alleles are inherited from the paternal genome in this embryo. With PGTM, most methodologies do in fact use SNP arrays, but they're relatively low resolution and low throughput. Our platform is a higher resolution methodology, so we can detect smaller imbalances and evaluate many more linked markers in parallel. But as I mentioned before, one of the big advantages is that we can test the mutation directly in parallel from the same biopsy, and this allows us to process cases without a trio or that are de novo, risk of de novo recurrence. Another illustration of the power of the technology is recently posted on bioarchives, where we've done the genetic analysis to show that you can actually eliminate whole chromosomes from the human embryo. In this study, we targeted a specific location on chromosome 6. This is an example of an embryo where the entire arm downstream of that target site was removed. And here's an example where the entire chromosome was eliminated. There are a couple of other studies that together have led people to review what's going on and consider that CRISPR may actually mess up embryo chromosomes significantly. But the other potential use of this is that we could prevent trisomy through the use of gene editing technology. So now with PGTP, you know, there's been several predictions in the past, including from Alan Handy's side, where he indicated that it should be possible to diagnose genetic predisposition to heart disease and cancer in 1993. In 96, Bob Edwards also suggested the potential for polygenic disease analysis in embryos in the near future. And our very own Jacques Cohen, who invited me to the talk today, also predicted 20 years ago the ability to use microarrays and predict predisposition to several complex diseases. Now, if we look at the actual utility of polygenic risk scoring, one illustration is shown here for breast cancer risk. You can see that, for example, individuals who are evaluated and found to have the top, or fall within the top 3% of genetic risk from their DNA alone, their lifetime risk of having breast cancer is significantly higher than individuals with a lower genetic risk. The real advantage here is that you can do this DNA testing early on, so before manifestation of disease, and potentially stratify patients to more careful monitoring if they fall into the high-risk category. And this has drawn interest from various economic groups. The Economist reported on this because there is a great potential to better utilize resources for human health. And so this was an example of breast cancer. Another is coronary artery disease in men. And so what we can do is actually the same thing in the embryo and identify embryos that are in the highest risk category so that patients have an opportunity to use that information to better choose which embryo to transfer and reduce the risk of disease. What I've shown so far is the ability to do this in the adult, and there are several publications, including one from our group, showing the ability to predict 16 complex disease risks, and another group who showed the ability to identify individuals with polygenic disease risk equivalent to monogenic disease risk. And there's been a real explosion in the field in terms of publications around polygenic risk scoring. Our group has actually taken this and looked at applying it to the human embryo. Our initial publication involved validation of the analytical performance. We demonstrated clinical utility by looking at siblings, and then developed a ranking system, which we call the genomic index, for patients to utilize this in the clinic. One of the most important results was to demonstrate that genotyping in the embryo is equivalent to the accuracy we can obtain from an adult. So from a trophectoder biopsy, we get equal accuracy as you would from a blood draw from an adult, for example. And so what this allows us to do is then look at how this might perform when we have adults with known disease status. And in this study set, we had over 11,000 siblings to evaluate. And you can see the prevalence of disease is known to be about 11%. Now, what we can do then is look at what happens if we randomly choose one sibling. The prevalence stays the same, 11%. But if we use DNA from these individuals and perform genetic selection, we can reduce the risk from 11% to 3%. We refer to this as a relative risk reduction, and the example, this is 73%. Actual performance is shown here for several common diseases, all of which demonstrated significant reduction in disease risk. And again, this is in a situation where there were only 2 siblings to choose from to begin with. I want to go through a couple of practical applications. In this case, the couple had a child, an existing child with type 1 diabetes, and was interested in using PGTP to evaluate their embryos. The testing includes aneuploidy screening. In this case, all 5 embryos produced were chromosomally normal. And then we were able to provide genomic index scores so that the patients would be able to select an embryo that had the best chance of being healthy. Another example is where, in a particular case, the couple elected to use PGTA. And in that process, they produced 5 euploid and 1 aneuploid embryo. In the genetic counseling, we found out that the partner actually was affected with type 1 diabetes. And so they had the opportunity, because they had used our testing to begin with, to simply provide saliva swabs to allow us to analyze parental DNA and then obtain genomic index scores for their embryos. With type 1 diabetes, we actually have a nice data set, which we published on in Frontiers in Endocrinology. And what we found is in families where there were up to 5 siblings with known disease status, the relative risk reduction of genetic selection versus a random selection was 72%. And so you might imagine that in these 2 cases where there were 5 euploids, not all euploid embryos implant, and so they may be in a situation where they have 4 euploid embryos available, and we still have a 70% reduction, with 3 embryos 55% and 2 46.7%. As part of a clinical trial, we also evaluated the average number of embryos produced, and on average, patients made 3.2 euploid embryos, 5.6 total, with a 59% euploidy rate. Of course, these numbers depend a lot on the population and on embryology laboratories throughout the world. but it does suggest that on average there are sufficient number of embryos to incorporate PGTP based analysis. This is our disease list, which we can categorize into 4 areas: diabetes, heart disease, cancers, and our first neurological disorder, schizophrenia. One concern has to do with whether or not selecting against one disease might enrich for another. And it turns out in a recent study, it was found that the risk predictors really don't overlap. And so with the genomic index, we've also actually found positive pleiotropy, so that looking at, for example, coronary artery disease is impacted in a positive way by also looking at things like high blood pressure. Another concern has to do with selection based on genetics as opposed to morphology. And it's relatively common practice in PGTA for patients to have information on the sex. In one program, with about 1,500 single embryo transfers, almost half of the patients chose the embryo based on genetics as opposed to morphology. It turns out that this is acceptable. It's really only recommended that each IVF program has a policy and explains the policy to their patients regarding sex selection in the United States. But more importantly, from this study, they also observed equivalent success rates, whether or not patients chose an embryo based on morphology or genetics. And so we think that this may be applicable when using PGTP to rank embryos for transfer. Another concept that may be important is that morphology-based selection itself, choosing embryos with faster development may actually lead to poor long-term health. We also know that patients with infertility already have an increased risk of cancer, diabetes, and heart disease. And so it may be that this population could benefit more than the general population, and that they already have a higher risk for the things that we can evaluate in the embryo. If we look at the genetic disease prevalence, monogenic disorders, for example, about 2% of the population are expected to have a monogenic disorder. The risk of several complex diseases is shown to the right and generally higher in the general population. If we actually look in situations where the embryo has a first order relative affected, that embryo's risk is actually higher in general compared to the general population. The ASRM has released an ethics committee opinion that suggests risk reduction of adult onset disease is ethical for arguments related to reproductive liberty. So essentially, patients should be able to make this decision for themselves when considering options that are available. They also recommended genetic counseling as an important component to this, and it is included with all of our testing. It's actually mandatory for PGTP, and as I mentioned, it is led by our head of genetic counseling, Jennifer Eccles, who has about 20 years of experience and has been a critical resource in the development of these methods. In general, we think that patients using oocyte donation who have many euploid embryos are one group that would benefit from utilizing PGTP. Any patients that are using PGT with our platform can add polygenic screening at any time. And also, we think it's an important option to make available specifically in patients with known family history. So if you had a patient come in interested in PGT and they told you that they were affected with something that we could test for, I think it would be important that they knew about that option. In conclusion, I've shown that our platform LifeView does actually improve PGT-A, M, and SR, and this is really through the combination of genotyping and copy number analysis. Polygenic risk scoring is in embryos is accurate as adults. We've shown clinical utility through genomic index-based selection amongst siblings with known disease status. And as I suggested, that PGTP is ethically acceptable, with these reproductive liberty arguments. And it could actually be said that it might be unethical not to offer this option to patients. In the future, we plan to expand our predictors to more populations and diseases. And we're launching a new clinical trial called the Embryo Health Test, where any patient that is using PGT for aneuploidy can add on polygenic screening. And our intention is to monitor patient perspectives over time with the utilization of PGTP. I'd like to thank Genomic Prediction, and the people that I didn't mention in the beginning include our computational biology team, our molecular genetics team. I'd also like to thank students from Rutgers University who've helped with genetic counseling development, our colleagues at Reproductive Medicine Associates of Connecticut, and for the gene editing work, Dieter Egley's group at Columbia University. And then, of course, the patients and laboratory partners and providers that we have with Genomic Prediction. I want to close by sort of promoting a new editorial. I put on my Jamie Metzl hat to talk about what might happen in the future if we are able to use IVF and genetic testing to significantly reduce the burden of disease in humans. And I also want to take this opportunity to ask all of you out there that are conducting research on human embryo genetics to consider submitting your work to the new journal Fertility and Sterility Science. Thank you.
Thanks, Nathan. That was incredibly interesting. It's really powerful data that you're showing there. And actually, I think it's a lot of it is context dependent. I think you're right in what you say about whether we're doing this or whether we're doing PGD or whether we're doing PGD for monogenic disorders. There's an undeniable overlap. It's just what people consider acceptable in their realms. And I think a lot of that is the delivery in which it is, in which a product is presented versus a clinical practice. So we're going to ask a few questions. I actually had one question for you while we load the other questions from the audience, and that was, what do you see as a baseline, what do you see as a low enough crossover between genetic interactions to be deemed safe? And what I mean by that is, you said that there's actually relatively little crossover between the genes. You showed the linear graph which displayed that actually we don't see that a mutation in one gene has much of an effect on another gene. And yet the global media headlines were dominated by the fact that when He Jiankui mutated CCR5, we saw that these people had an increased risk of West Nile virus, that they had a potential for life-limiting, based off the UK Biobank certainly, that they had a shorter life expectancy. What is your, I guess, how do you address that when you talk about how we approach a bare minimum for predicting crossover?
Yeah, I think there's certainly some good examples of where pleiotropy is in effect. So, you know, the hemoglobinopathies, for example, and what you mentioned, and typically those are easy to define because it's a single gene. But with polygenic disorders, right now it turns out that there, again, as I said, there's not much overlap in the predictors for sort of unrelated conditions. And actually in situations where there is a relationship, you know, coronary artery disease and high cholesterol, for example, actually benefit each other by having one predictor. It reduces the risk of the other disease. So, we're really just learning about this now that we have these polygenic risk score algorithms, and the paper that we just published is probably one of the first to really look at it in depth.
Again, this was based on the UK Biobank, right? This is the sibling data.
Correct.
Yeah, really interesting read. Okay, cool. We will.
Well, I think we can start fielding some of the questions that we've been receiving in the chat. There have been several in the area of equity and access, and maybe to Jamie and possibly Helen, if you want to editorialize afterwards, is how do you address the public perception or expectation that art technologies will widen the gap between the haves and the have-nots and promote inequity, in essence, fostering a genetically superior class of people who dominate all others? Now, this is sort of utopic, but imaginable. How do you sort of defend that question or that position, Jamie?
So certainly I'm not trying to defend it, but that's certainly one possible future, unless we have the meaningful conversations that we need to have now and we build the infrastructure so that there is equitable access to these technologies. Certainly every technology has its early adopters, and that gives them an advantage, and whether that's the stirrup and the Mongols and everything else. So we can't be against first adopters. We need to make sure that we have equitable regulatory and healthcare systems that can share the benefits once we're clear what those benefits are. And certainly I praised the United Kingdom, Israel. There are other places that are trying to do that in a more thoughtful way than others, like in the United States, where people with money have more access. And that's why, as I was saying in my remarks, it feels like small personal questions, but they're connected to big societal questions. And the issues of equity and diversity, that's not an afterthought. That has to be woven into everything that we do and think about now.
Excellent. I think I agree with that. Helen, do you have anything to add to that?
No, I think equity comes down to it. I don't think that, we can always blame a role of genetics on widening the gap between the two, but actually common healthcare systems and the access to care, no greater thing will reduce your lifespan than lack of access to decent medical care. And unfortunately, this is an age-old phenomenon where those with money will receive care sooner than those without. I think that's a better one to address.
Yeah, could I just ask Jamie with a follow-up? What do you think the buy-in is from governments and insurance companies? I mean, we're talking about things that are on a long-term scale.
So right now it's relatively small because this science is experimental. I mean, I loved, Nathan, that your presentation was delivered in kind of a dry manner, because that masked how unbelievably revolutionary the topic that you're discussing is, not just for healthcare, but just for the future of humans on Earth. And so there's going to be first adopters. And I mean, Helen was very clear that in terms of practical applications, we're just at the very, very beginning of this journey. But I think that over time, for the reasons they articulated earlier about both cost savings, reduction of pain and suffering, and basic desires by prospective parents, once we pass that safety threshold and a societal threshold, I think things have the potential to move quickly. And that's why, though, it's incumbent on all of us to anticipate the kinds of questions that everybody's going to have, like in the first one of equity and diversity, and just make abundantly clear in everything that we're doing that our goal is not some kind of horrible eugenics, but to actually solve problems and help people live healthier, longer, more robust lives in a thoughtful, sensitive, and inclusive way.
Thank you, Jamie and Nathan. Maybe a question for Nathan that seems to be percolating in the questions here is about numbers of embryos. Nathan, you mentioned in your talk that candidates for some of this technology today would be patients with high numbers of embryos, but the hope would be that this is more sort of, applicable to a greater range of IVF patients, maybe all patients going through treatment. At what point do you think there's a loss of the value proposition or Is there a minimum number of embryos? I mean, this is true. I've been interested in embryo selection, embryo ranking, not genetically, most of my career. And, you know, when you have 1 or 2 embryos, your power goes down, goes away. So how does this sort of play out with genetics and genomics?
Yeah, I think actually it's pretty simple. You know, you have to have more than one, right? In the data set I presented, we had over 11,000 sibling pairs where you only had 2. And there was a significant relative risk reduction. So, you know, you can offer it to every patient and they have an opportunity at the end when they know that they have more than one euploid embryo, they can decide at that point if they want to add it. And we have shown that there is a significant benefit to including the information in the decision, even with only 2 euploid embryos.
And I would imagine even if you have a single embryo and you have some sort of informative data about the trajectory of that embryo as it becomes a human being, you may be able to do some lifestyle changes or, you know, pre-treatments during pregnancy to sort of alleviate or diminish the effects of that mutation or that condition.
Yeah, I think a lot like the example with breast cancer, having that high risk prediction earlier on is important. It's kind of the concept of forewarned, forearmed. And patients may want to have that information even when they do only have 1 embryo. And some patients may not. And there's a lot of reasons they might have to make that decision. It's one of the reasons we're doing this observational study, to really get more information on patient perspectives so that we can help more people in the future.
I've one more question until I give it over to Helen to ask a few or manage some of these. And that would be maybe to all 3 of you. How long do you think we are going to have to wait to have a finger on the pulse or a proxy on how we're doing with our selection in real life, in actuality? Decades, generations, what do you think?
I think it's actually much closer than we think. Always, the last time I said it's in the next 5 years, it happened that the next day I flew out to Hong Kong. I remember giving a talk in London saying, you know, this is probably being already done in China. And then I was accused of knowing something on the inside. Really, I think we often project 5 years, 10 years, and so much of this is an iterative process. We're constantly redefining, we're constantly reassessing things, and it's less of an evolution, or less of graduation and more of an evolution, that these things kind of slowly creep up and happen. I think with the incidence of our ability to detect using less invasive means, I think that is going to be the main game changer in terms of where people's limitations are. So being able to determine more about an embryo without having to disturb it, being able to determine what maybe we could do with that embryo, and maybe then the only disruption you would create to the zona would be to correct a mutation that you knew was there.
Yeah, and what I would say, last year I was in Kyoto and I visited with Michinori Saito, who's one of the world's experts on in vitro gametogenesis. And I asked him exactly this question: how long do you think it's going to take before we know it's safe? And he kind of leaned back and he said, well, I'm thinking about 240 years, 3 full generations. And I'm sure if we were a more patient species, that would be about right. But as Helen just said, the second there's the capacity to do some kind of thing, even if it seems crazy to a lot of people, someone somewhere is going to do it and either prove it or disprove it. 100 years ago, we had less than 3 billion people and a 20% literacy rate. Now we're almost 8 billion and an 85% literacy rate. So just the number of people who are part of this process of innovating, of trying things, is just so much more. And I spoke earlier about the superconvergence of all of these technologies. And so there always will be innovators like Nathan and genomic prediction. But once something is proven, I mean, using the He Zhonghui example, had He Zhonghui succeeded, we would be in a very different place now, even though everybody was such a critic of how, myself included, about how unethical it was. If Nathan Treff and Steve Hsu and genomic prediction, if they can start proving that, that doesn't mean everyone's going to do it at once, but it means that other people are going to start replicating, and then other people are going to start replicating that. So I think that it could happen pretty quickly, but I agree with Helen. It's not going to be oh yeah, today it happened. It'll just be a little more every day on an accelerating curve.
I think we also have geographical bias on these things. If that had been done in the United States or in the UK, people would have been possibly more accepting of it. But actually, there's this fear of, you know, the Wild West of the West, or not the West, of the East, and that unfortunately is unfair on researchers there.
But what I would say, pushing back lovingly, is had it been done in the United States or Europe, it wouldn't have been done in this way. And
no, very true.
It was done reflected in part the scientific culture of China, which is part of the problem.
Agreed.
Great.
I'm looking for some more questions for everyone. So the internet has proposed that genome editing technology proposes or promises to cure diseases and make us stronger and smarter, but where do we draw the line? And I think alluding to, I'm going to take the question myself actually, alluding to what you said about the infrastructure in the UK and our incredible legislative system, which has a governing body that resides over all research, including gametes. And this is reflected on, or I think respected globally. I think that is the line drawer. I think the line has been drawn for us by the lists of genetic conditions that we are currently allowed to screen or use for PGD. So very often when this slippery slope argument comes up, I think people forget the fact that there are laws in place. So I presume you both agree with that.
I do, but I will maybe add to it. There are no lines. It's silly for us to say, I mean, there are norms, there are things, and those norms are always changing. And that's why I'm totally against the slippery slope argument, because it's like, you know, imagine we're a bunch of monkeys climbing in the trees. Like, we can't climb down in the trees. What's next? Slippery slope. We're going to be walking on 2 feet. I mean, it just, there's no norm. It's an evolving process, but we can't get ahead of ourselves. We need to have systems and structures that make sure we're going where we're going together, that we have a set of values, and these are in many cases are ancient values, and that our processes and procedures are living up to those values, that we're not dehumanizing ourselves. But who knows where we're going? I mean, over thousands of years, these simple things that we're talking about today will, I'm certain, fundamentally revolutionize who and what we are as a species in ways that we can't even imagine. But we don't want Nuremberg-style human experimentation. We really need to make sure that we are preventing the abuses as we define them, and then we need to embed those values in whatever our systems are, whether it's the Universal Declaration of Human Rights or our theological traditions or other things.
Well, thanks, Jamie, and thank you, everyone. I think we're getting close to our time for ending, and I just wanted to commend everyone for being involved in such a dynamic profession and professions. I think we're very lucky to be involved in such an intersection of such a powerful powerful fields where we have, you know, computational genomics, bioethics, molecular biology, you know, embryology, you know, all intersecting to potentially change the world as we know it today. So from being in a motorhome outside of Indiana, and from your hosts in the United Kingdom and speakers in the United States, other places, I want to thank you for your participation. This has been a great enlightening session, and I look forward to seeing you all on the next i3 webinar. Have a good evening or good day wherever you are. Bye-bye.
Thanks so much, Barry, and thanks for being a co-moderator in this session. I'd like to encourage all of you in the audience to please contact i3 if you would like to help create or submit some website content through our website if you have any ideas for partnership. Also, so far over 15 embryology and science organizations have pledged their support. Please remember all the sessions have been recorded, so they will be available tomorrow morning for all time zones. Thank you to Thomas Elliott for arranging all of this. And now for closing this session and making some important announcements is our Irish embryologist, our fellow Irish woman, Anna McLaughlin, who will be closing the session. Anna, over to you.
Thanks so much, Helen. Yeah, so basically, I just wanted to introduce the social media channels again, and basically, sorry, I'm just trying to get my slide up here. Yeah, there we go. So I just wanted to update everyone that we do update our website and our social media channels regularly. We update them with the dates of the meetings and the content. There you can see the slides on the handles for our LinkedIn, Instagram, Twitter, and Facebook. So please follow for regular updates. We would also love any feedback that you can give us. Talks that you've enjoyed and ones you'd like to see similar topics off again. Or of course, we're also welcome to hear of any new topics which you would be interested in hearing in the future. And I would like to introduce next week's session, which is going to be on Tuesday, August the 18th. So it's the same time, and it's going to be the 29th session, and it's on cryopreservation. Our first speaker will be Dr. Jeremy Chang, and they'll be discussing principles of cryopreservation and optimization of vitrification. Dr. Anna Kobo will be discussing the state of art in vitrification, the best technical approaches and outcomes. And finally, we'll also have Professor Anja Pinburg discussing the health of offspring following frozen embryo transfer. So we look forward to seeing you all there next week, and thank you so much to everybody for joining today.